So now we are about to move on to the command line intersection. Now that you guys already know about R, how to install R Studio, different libraries and how to use these libraries, we will now move on to analyzing the data just like we did on graphical user interface to command line interface. But first of all, we need to get around the data. We need to little know about Bioinformatics Microarray tutorial. We have to get the tutorial as well. So what we are going to do? So overviewing our whole plan. First of all, we are going to get the data. After that, we are going to apply the QC on non-normalized data. What is the QC? QC means quality control. We are going to check the quality of our data, of non-normalized data. After that, we are going to do the normalization. So after checking the non-normalized data, we are going to normalize the data. After normalizing the data, we are going to correcting its batch. For example, we have 50 samples of normal data, 50 samples of thyroid data. We can use it to batch correct the sample. After that, we are going to remove the outliners, and then we are going to apply the quality control on normalized data as well. Then we are going to do the covariate analysis. After that, we are going to annotate the probes and then collapse the rows, and after that we are going to get our differential expression analysis. That is how you get your DEGs, which are differentially expressed genes. So first, what we are going to do is we are going to get the data. How we can do it? So moving to the code section, here we will move to our R Studio. Here you can see we have this beautiful layout which is saying to use the bioconductor to download the libraries. I think you already know that. So first of all, what we need library IFE, which is a library that is used for Bioinformatics Microarray data analysis. Then we need the GeoQuery, which will get the data from the database of Geo. And then we need the Lima package to annotate the data. Then we need WGCNA to create the dendrograms. WGCNA is the weighted graphical correlation network analysis. Then we need the SVA to plot the graphs. Then we need the Biomarker and then the Biobase database to analyze these data sets. So first, what we're going to do is we are going to load the libraries. So let's just do that. Load the IFE library, GeoQuery library, Lima library, and WGCNA library, SVA library, Biomarker library, and your Biobase library. Then we are going to set the directory. I think you should be familiar with setting the directory, because what we need to do is, for example, we have this microarray course, and first we are going to just copy this, and here we can see, if I am gonna show you how to set your directory, and then just paste the directory here and then remove your backslashes to forward slash. That is how you include how to set a directory in R. So that's pretty much it for setting the directory. Then we are going, what we are going to do. First we are going to get the data, then we are going to quality control on raw data, then we are going to normalize the data, then we are going to batch correct the data and things like that. That is all the comments, so you don't have to worry about that. So what we need the Geo dataset. That it is there are two choices for getting the data. You can download the data from Geo database. For example, you can copy the accession, go to your browser, and after going to your browser, you can just open your geo database, and after opening your geo database you can search for the accession number. Here I will search for the accession number, and then I can download this raw data, which is 236 megabytes. But if I'm not gonna do it, you can also run the script to get the data. After getting the data, we are going to annotate it. I'm not going to tell you how to write a code because this code will be available on my GitHub profile. You can pick up from there and use according to your own need. So that is it for this coding section, because you should be familiar with how to use R. I'm just here to show you how to do the microarray analysis and how to get the perfect graphs for your research papers. So you can run all this script. Then we are going to get the geo supplementary files. That will get the .clf files to create the IFA object with the expression data of entire experiment. That will be your slice. So we are going to trying to get the URL from here. You can also see we are downloading the file. So it will take a lot of time to just download the files as well. So for that we are going to move to our presentation. So here you can see we got the data. After getting the data, we will apply the quality control. So what is the quality control? Here you are seeing a box plot. If I can laser point it, you are seeing a box plot that is pre-normalized. You are seeing intensity of array and then the size of array. Control is your control is shown by the black curve, and PKD is Parkinson disease. So this data set is based on Parkinson disease. So if I can show you that as well. So if we move to here you can see the transcriptional analysis of multiple brain region in Parkinson disease. So that is how you got your data. And we didn't have the permission to get the data spliced, but that is then you don't have to worry about that. Error in GSPLICE, failed to download. In addition, cannot remove file. So reason permission denied. So it didn't ask for any permission at first, but you can move on if you want to and check the CLI files. So it will not allow us to get these files because we haven't got the data yet. So what you guys can do is you can get this data and then extract it and then run your code and download your data to your local computer server before loading it into R. After that you can get the phenotypic experiment, check your reordering, reformat it, then apply the quality control, and then just check it by using some methods. So that is the prehistoric technique of getting the differentially expressed gene. But now that we have got our we know how to get the differentially expressed genes, the main thing is now to analyze that data. So I'm just gonna show you here because it's gonna take a lot of time and you guys can't wait for the data to be analyzed in pretty much that late. So now that we have the box plot to ourselves, here you can see we have the box plot of control and Parkinson disease data. Then we will apply the, here you can see the history of log expression as well. That is the quality before the quality control. Now we will batch correct it. Now we are correcting the post normalization pre-batch correction and the MDS post batch correction as well. So here you can see the control and Parkinson disease points are now in the middle, so they are contributing. So we will remove the outliners. These are the samples that are included. When you will download these samples, you will get to know around these accession numbers, geo accessions as well. Now we will apply the quality control on normalized data. After applying the quality control, you will see a big difference that all the data is now normalized. Here you can see it is not normalized. Now after correcting it, the data is now normalized. Expression density is the same as I showed you on the graphical user interface, and it is also involved in MDS. So now we will do a covariate analysis, which will be the whole data analysis of what age of control and Parkinson disease is used, what badge was used, what male and female difference subjects, and then we will move on to annotating the probes. This is all done by your script, so you don't have to worry about that. After that, we have to collapse the probes and get the differentially expressed gene that will show us the p-value. So that is the process of getting the differentially expressed genes. So now that we have the differentially expressed gene in our geo databases, how we can use this databases to get the perfect plots for that. So for that, we need to do is we have to do the microarray analysis, which I can say is basically about plotting some good plots of volcano, heat maps, and things like that. So what we need to do is we need to download our dataset, or you can just run the script and it will get the data. And then load the libraries, ggplot, multus, and then mass library. Read the data in annotation file. You can set the directory as well. I already set the directory, and then I'm just gonna read the data that I have downloaded. So for this specific code, what you need to do is you need to copy the link and go here and then download the data file. So the data file that you need is GSE35570 series matrix.txt, and that series matrix.txt. Now you should remember that it is based on your thyroid radiation data and your thyroid data, and after that we just need the file of annotation text and series matrix.txt that I am going to show you. Here you can see the series matrix file, and here you can see the raw file that you need. So after getting these files, you will calculate the Pearson correlation value. These are the algorithms that I showed you on the graphical user interface that is used for the differentiation of differentially expressed genes. So now we got applied the algorithm, then we will check the correlation between them. For checking the correlation, we will use a heat map. You can see all type of correlation metrics and for cancer samples and things like that you supposedly want to get. So that is for getting the CV and the mean plot and things like that is just for plotting some good plots. So here you are getting the outliners, column names, your theoretical cancer and the mean plot, gene IDs, CVD IDs, and things like that. So all the values that are generated for this plot are represented here. You can check this by attribute names, things like that. So for now, moving on, we will want to filter out the genes in the bottom quantile of the means. So we can do that as well. Then we will move to ANOVA and return the p-values. After that, we will adjust the p-values and the data in it. Now the data is now being adjusted and the adjacent p-values will be given to you. The p-value, first we get the p-value, after that we change it to the adjacent p-value. So now the script is being run. You need to have a good computer to run this analysis as well, because I am using a 16 GB RAM, a graphic card which is of 4 GB, and a 11 to 12 gen processor to run the script, but it is taking a lot of time as well on my system. So if you are using 8 GB of RAM or 4 GB of RAM, or you are using MacBook, that is okay. MacBook can run these type of script. If you are using that, you can also check Google Colab to run your R codes. Now the script is run. Now we got our adjacent p-values and the row names. Now the significant genes and the subset of data metrics are now being plotted. Now we will apply the principal component analysis, which will be the PCA. PCA will plot the data and we can get these data and the heat map and all this that is required. So if we here we will see that here is the data file that we are using, and this is the annotation text file that is required for the sample. And now I want to find the most upregulated between the class and calculate the log fold change in it. I will to do so, and then I'm going to mean it by the theoretic cancer and the normal cancer means, and then print the 5 most up and down regulated genes. So here you can see here is the top 5 up regulated genes, here is the top 5 down regulated genes. You can get the idea by the values, and after that we will plot the expression folds by using the volcano plot that we did in the graphical user interface. We check the volcano plot, and then we will apply some classification using the linear discriminant analysis and get the data trained as well. Now the data is being trained, we will return to the point color based on cluster assignment. Here we will get our databases, and now we can see that these are the genes that are available here, and there is a error that is saying we cannot closure the type effector. So one thing that I noticed that we haven't come across any type of graphs. So why I was thinking it is saving the graphs. So this is the final file that we received. So we have the gene. So let's search the gene that we got from our GUI based analysis. So that gene was PRR15 gene. So let's see if it is included in all the samples file. So here we can see we got our PRR15 sample file. So let me just close this. If I want to run it again, why it haven't plotted anything? So I think there is a problem with my Okay, I think there is a problem with my R Studio because it isn't plotting anything. So I'm just gonna restart it and then see what happens. So if we run the code again, let's just see. And ggplot, and now we have this library, and after getting the library, can we, okay, so we haven't set the directory, got the data, so let's just get it again. Why haven't I showed you any plot? So I'm confused for it. So let's just do it again, annotate the data, apply the algorithm and see. So sorry for that, my RStudio got stuck in it and it didn't show you any plot. So here you can see correlation matrix of thyroid cancer samples. So if I zoom it in again, here you can see, you can't see anything specifically, these are just the sample files that we just studied. So now we are going to analyze, we are going to annotate it and apply some sample files, and you can see we have our genes and samples now being analyzed, which is the sample CV, which is correlation factorization and your mean values. And after that you can apply the gene of the mean, gene ST value, gene CV value, and do some plotting against that. Here you can see this. So that is for applying the data and getting your values again. So I'm just showing you to do this again. So adjacent p-value is going to take some time because I want you guys to know that RStudio can be stuck sometime. So if my RStudio got stuck, yours can be as well. So what you need to do is you need to run the script again if you want to do the significant changes according to your own needs. And now we are getting the plots. You can significantly see these plots are much different than the plots that you got in your graphical user interface. So here you got the freedom of getting the plots that you require for yourself, the plot that you need for yourself. So you can also change the color scheme. You can also change the values of the genes. So that is all totally dependent on you. So I think now it is again doing the adjacent values. P value is now being adjusted again. Now I want to show I want to specifically show is the PC analysis. How we can analyze the data in PCA. So here you can see that we have the PCA plot of pulmonary thyroid cancer PCA1 versus PCA2. So in the PCA1 contains the radiation exposed sample, and the spotted cancer contain the red and the normal files. So you can see that the normal files were apart so less expressed than our cancer files. So now we will move on to the heat map and then just plot the heat map to see what it is going to give us. So the heat map is according to the legendary color scheme that are being used, red, black and white. And now if you want to find the most upregulated and downregulated gene, you can do that as well, that I showed you previously, the normalized and non-normalized data. So here you can see the heat map is now generated. So I will zoom on into it and show you. So now my laptop is getting really hot. So you can see these are the genes, these are the clusterization of your genes versus your samples. So these are the top expressed genes and these are the samples that are given it. And here is the expression. Upregulated is shown by green button and downregulation is shown by red. So you can, I think you can pretty much see what it means. So that is it for your heat map of getting your radiated and cancer radiated data. So what we want to do is we want to print out some values and apply the volcano plot on it. So here you can see the volcano plot of normal versus tumor sample folds. So it is now being customized and the top regulated genes and the cancers and the cell line and things like that you can see. So you can zoom into your volcano plot and see the genes that are being expressed. You can also change the color scheme in here and you can do pretty much what you want to. You can get a table of the data and after you can also return the color point and plot the first two discrimination of your PTC. So here you can see that is the analyzed data file that you need. The plot are being saved in your R as you continue your analysis. So that was pretty much it for how to do the analysis using R. So there is another R script that I got ready which is quite small for it. So what you need to do is it is based on the preliminary fibrosis differential expression. So we are going to do this quickly as well. I'm just gonna clear all the plots from here, clear all the history and then load the packages, get everything done and retrieve the microarray data and store the first dataset, just the first dataset, and then make a matrix file that will be named GSE and get the data. So this data set is now based on cancer. It will be your control against cancer. So what specific cancer that we are dealing with? Let's check that while it downloads the file. So we will move to geo accession number and paste our accession number and see. It is a molecular phenotyping of idiopathic intestinal phenomenosis, which is also a type of cancer. So now that our file is here, we can create a matrix called GSE and then get the sample information and annotation and plot. So check if the data is normalized. So distribution should be familiar. So how to check if the data is normalized? You can see all the data that is presented here are kind of point to same. So the data is normalized. It is just the same. One or less two value doesn't make any such difference. And if you want to export these graphs, you can export them as an image and export them as a PDF and things like that. You can also copy them to clipboard, copy plot, and then move to your presentation file that you make for your research work and then apply the paste the plot. So that's how you can do. So there is some other thing that you can do as well. You can create the heat maps and here you can see we created a heat map of our control, our COP, DIP and the sample rates and the expression of a significant sample that is highly expressed. So I hypothesize age. So I was working on age when I was working on this data. So create the principal component analysis. PCA is important for your microarray data analysis, so we must include that as well. So we are now working with the 80 samples. 80 samples are included in it. So here you can see the distribution of your samples. The control are shown by the pink color. COP samples are shown by this, DIP this, and you can check all that that you need. I will upload all the data on my GitHub profile. You can create the model and rename the column, create the median level, identify the gene expressed in 2 or more couples, then assign the gene, get the genes, and annotate those genes, calculate their relative assay weight, also calculate their construct, their weight matrix, add the gene symbols, then get the results, create volcano plot that will be labeling 20 genes and that will be turning significant result, and the volcano plot will be based on this. And so here you can see you will get your volcano plot. Here you can see the top regulated gene, the low regulated gene, and then you can print the top significant gene. So here you can see the 20 top significant genes are represented here. So that is pretty much it for how to do the data analysis of if a matrix or some other kind of technique that is used in microarray data. You can use those to do the analysis. So that is it for this section of the course as well. How to do the command line interface used to analyze the microarray data and get the perfect plot using the RStudio. And I think this should be enough for this. I shall see you in the next section, or maybe I'm gonna conclude this here because there is there is nothing more to advance in microarray data analysis. You got your plots, all the scripts that are used in this course, all the data that is used will be provided in the code and will be available on my GitHub profile. Thank you so much for your attention, and using that you can also approach me on my GitHub repository, and also can approach me on my website if you want guidance. If you want to do this analysis and you're getting stuck somewhere, I will also try to help you, and you can also reach me in my Udemy inbox, or you can start a discussion with another students with me. I try to answer your question within a day. So thank you so much if you are learning this course till so far. I am grateful for you, grateful to you, thankful to you to be a part of these courses. And that is pretty much it for the course, and I will see you in the next one. Thank you.